Yes, we working on that part.
Cool, nice. That's normal.
Okay. All right, good. So the recording has started. So the idea is that you have 3 regions. No, you have 6 regions. 6 regions. Each region has 3 locations.
Mm-hmm.
And when you do the observations within each region, you get different results for each location. And that's due to 2 things. One is due to the observation error, and the other is due to the variation between locations. Because even in one region, there may be difference between locations.
Yeah, that's clear for me.
Yeah. So the whole idea is to try to estimate how much is due to the observation error and how much is due to the difference in location.
Okay.
So to do that, we need Bayesian statistics, Bayesian model. And the idea is that C is your observations,
mm-hmm.
and Q are the parameters. So like mean, variance, and so on.
Okay.
So we have observations, and we want to estimate the parameters. So we want to get the observations. We want to get Q from C. So we have C and we want to get Q.
Okay, yeah.
So this formula will help me get Q from C.
But that is the Bayes formula, yeah, I understand.
Yes, Bayes formula, yes, okay. Now we can compute this. We can compute the probability of C from Q. And then we make some assumption about these. It turns out the only assumption you need to make is that this one divided by this one is a constant.
Yeah, of course. Because C is our data, and Q are the variables.
Q are the variables, yes. Q is what you want to fit. So to do the fitting, so this is just standard expression for, let me see here.
Why is an integral?
Oh, because it's a continuous probability distribution. For instance, if you have a Gaussian, the reason it's an integral is that to find this probability, C actually depends on another set of parameters called lambda. And lambda is a continuous parameter.
Okay.
So if you have a continuous parameter and probability with a continuous parameter, you always have to use an integral. I mean, you have seen this kind of thing if I take if I want to find the probability in the blue, right? That's an integral. That's going to be an integral. It's an integral of this function.
Yeah, it's a question. It's an integral because we need like a measurement the area of the data.
Yes, it's the area under the probability density.
Okay. Yeah. That's clear for me.
I mean, if you take here, if I want to find the, I'm not sure if they will show here. And every time you want to find a distribution, I guess they don't show here. Let's see if I take this.
Protocol later into
I mean, for instance, here he has an integral because he's trying to find the probability of being inside some area. So you always need to do this when you have continuous probability distribution.
But with this integral, we can calculate all the area in which is distributed our data.
Well, it's not calculating the integral doesn't calculate a area; it calculates a probability.
Okay.
It's calculating a probability.
Yeah, and what means the equation 2?
So this is saying that if I know what the parameters are Q, then I can find the probability of C, and C is my observation.
Yes.
So I have a model, and my model has the parameters Q. If I know what Q is, I can find the probability distribution for C, and C is my observation.
Okay.
This is something like, for instance, if I take, if you look here, I'm actually using an exponential distribution. I don't know if you're familiar with this, but you often do this with exponential distribution with population. But you can see that for the exponential distribution, the probability is given by the area.
Yeah, I understand. And for this reason, we are using integral.
Yes.
Yeah, it's clear for me. Okay, nice.
All right. Now we actually have 3 different measurements.
Because we need to obtain the area now. The area. That's the idea.
Yes. Since we have 3 different measurements, we actually have 3 different integrals.
Why 3 different?
Well, we have 3 measurements because we have 3 measurements in each region. So maybe I can draw a picture here. Let's see if I can do this. Let's see. So let's take one location. Take one location. Okay, and then you think, and you have a data. You have a data for one location. You have a number. You have an abundance number. You have a single measurement because over time you take several measurements. So let me call that measurement. I'll call that measurement C1. Now C1 depends on the actual abundance. At that location. Right? C1 is a measurement. That's a measurement.
Okay.
So there's some error, but it depends on the actual abundance, which I'm going to call lambda. And then I assume, the usual way to assume this, I assume that the measurement has an exponential distribution
Why we are assuming that?
That's a common assumption. When you're doing population measurements, that's the usual way, that's the usual assumption.
But in this case, it's not really a Poisson distribution.
No, I'm sorry. I should say Poisson. Sorry. I should say Poisson.
Because it's a count, I understand.
It's a Poisson distribution. Sorry, not exponential. Okay. Poisson distribution. So that
makes sense.
Yeah,
more for me.
So that means that the
Okay.
so that means that the probability that says C1 is less than A or something is equal to integral from 0 to A of the Poisson distribution of I can say Z and lambda, and that's going to be dz.
The lambda, no?
Yes. So the lambda gives me the actual. So this is where the integral comes from. Now, the point is that lambda also depends, lambda also is different for different locations.
Why?
Well, because in some region here, even though it's one region, even though this is Bombascaro, right?
Yeah, okay.
You don't have the same abundance here, here, here. You don't have the same.
Yeah.
Okay. So this would have lambda 1, this one has lambda 2, this one has lambda 3.
Yeah, that means that is inside each place.
Yes, inside each region, there's a different
Okay, good.
rate.
Yeah, that's clear for me.
Okay. And we make the assumption, we'll assume that lambda is, so lambda also has a probability. Because it's not the same everywhere. And you chose some location, you chose the location randomly. So the lambda that you get from your location is going to be random.
Okay.
It's not known. This is not known. It's a random variable. So I'm going to assume that lambda is uniformly distributed between Q1 and Q2. So this would be the minimum. This is the minimum abundance. Sorry. And this is the maximum abundance. So if I pick up a location here, I don't know what lambda is, but I know it's between this number and this number. So the problem is to estimate Q1 and Q2
Okay.
from the data. I mean, here we said Q1 and Q2 determine the distribution of lambda. Lambda determines the distribution of C, but we have C.
Yeah, of course.
So you have to work backwards.
Mm-hmm.
So you work backwards. From C
Yeah.
to estimate Q1 and Q2. That's why you use Bayesian.
Yeah, I agree with you because with Bayesian approach, we can include previous data, you know.
Yeah. Yeah.
Cool.
Bayesian approach gets you the distribution from the data. All right.
Okay.
All right. So this is giving me, so you can recognize this is Poisson here. So because I have three locations, I have three Poisson distributions.
Yeah, because each one is different.
Yes, each one is different. And then here I have my Q1 and Q2. All right. And here I'm integrating over lambda because I don't know lambda. lambda depends on Q1 and Q2.
Yeah, I understand.
So that's where all these integrals come from. So that gives me this, I write this probability as a product of 3 terms. Each term looks like this.
Yeah.
And then this is just some mathematical stuff. So in this Bayesian expression, I can compute this, and then this one, I just need to assume that this is a constant. These are what you call prior probabilities. And since I have no information, we just assume that it's a constant.
Okay.
Now this is the usual way to do things. If you have more information, then you may make some other assumption. But right now I'll just assume that it's constant. So that's why I say here, when you don't have information, then usually the only assumption you can make is that it's a constant.
Yeah, but we have previous information and we can include. It's okay.
Yeah, but right now we don't really. But maybe we can get something. I'm not sure. All right, so this is mathematical. So
and that information is lambda valued, no?
So the information would be, do you know something about abundance? I mean, do you know that the abundance has to be less than something?
Okay.
Do you have do you have some previous idea about what the abundance is? And some previous idea of what the measurement is going to be. But if you're just going into the region and making measurements the first time, then you have to have some other information besides the measurements in this data set.
Yeah. No, that's clear for me. Remember that we had 3 observers, and the 3 observers move around all the randomly in all the region.
Yes. But the 3 observers always go to the same 3 locations, right?
No.
No, but I mean, you have 3 fixed locations, don't you?
We measurement the data at the same time in the 2 locations.
Yes, that's right.
Yeah, but the next time we randomly move.
Yes, I understand. The observer changes, but the locations do not change.
Yes,
the observer changes, but the locations do not change.
Yeah, that's true. And with this, for this, we try to dissipate the error in all the samples.
Sure. All right. So this gives me the information I need. Then I use this mathematics to estimate. So I have to show you the result. All right. So actually, I thought I did family, but I guess I don't have family yet. I think I didn't put family in the.
Yeah, maybe just we need to include the family data distribution in that plot.
Yeah. So this is showing you. If I make this bigger. So this is showing that, for instance, if you just look at species, almost all species have counts less than 100.
Yeah, of course.
Or this is not species. This is transect. So it's species and it's species of bird and species of fruit.
Yeah, that are transects because remember. But in this case, we didn't use the point counts data.
Didn't use the point counts. Yes, that's right.
Okay.
So you can see if I use a genus, then it's bigger.
Yeah, the genus is. But the behavior is very similar, no?
Yeah, it's somewhat similar. Still, you have a lot of counts that are very small. and not
true.
so many counts that are bigger. So let me...
Cool, I understand. That is really good. But what about that plot? What means that plot?
All right, yeah, I know. This is probably a little bit difficult to understand. So this is supposed to show show what the distribution looks like. So all right. So first, let me explain the X and Y scale. You have three locations, right? And so of these three locations, you have three total counts. For each species, you have three counts.
Yes.
Right. Now you have the highest count, the middle count, and the lowest count. Now, in order
why high is low with a mid?
Well, so I mean, since you have three counts, you don't know where the mean is. Somewhere in the middle. But you do have you have a highest count, you have a middle count, and you have a lowest count.
Okay, yeah. No, I understand. Okay.
Now, these three counts, it's hard to plot three counts in the graph. So what we do instead is you take the highest count.
Okay.
Well, let's say you take the lowest count and divide it by the middle count.
Yeah.
And that gives a number between 0 and 1.
Yeah, it's a proportion.
Because it's a proportion, yes. So this here is that proportion. Lowest count divided by middle count.
Lowest divided by the highest count.
No, by the middle count.
By the middle, okay.
This one is the middle divided by the highest.
Yeah.
Okay, so this is lowest divided by the middle, and this is middle divided by the highest.
Okay.
Now, we could do lowest divided by highest, but I think this is probably better. So what this is showing is so you have three observations, right?
Yes.
And you can take the mean of those three observations.
Mm-hmm.
So you would hope that the mean of those three observations would be close to the actual mean in the region.
Okay.
But it turns out that it's not. The mean of those three observations is always bigger. Now, if the three observations agree well, then the mean of the three observations is close to 1. So that means that if the three observations agree well, then the actual mean will be close to the mean of those three. But if the three observations are very different, that's not true anymore. It may be that the actual mean is much larger than the average.
Yeah, okay. Now, if we can find that point in the purple region, that means that each data will have a higher mean.
So it's not higher mean; it's the agreement. The agreement is better.
Okay.
You have agreement that's better. Now these dots are showing the actual data. So, for instance, this black square.
Okay.
Copalinga.
It's Copalinga, yeah, okay.
So that means that one observation was here. And you can see that there are many cases where you have where you have 0.
Pretty close to the axis.
Yeah, this is not good. This means that in some locations you had 0. You had no counts.
Yeah.
So in many of the observations, either you have 1 or 2 measurements that are 0, and only one that's not 0.
Okay. And the squares in the purple place.
So these are you can see that only Copalinga has good measurements. for species. Now, if you do for, that's only for species. But if you look for genus,
Okay.
it's a little bit better. If you do for genus, then you get some other places that have better measurements. This Copalinga, this Bomboscaro,
We have circles.
and the Finca. Finca has some.
Circles is one good.
And then this one here is ECSF. But Bella Vista,
Yeah, for instance, Bella Vista and Cajanuma we can find.
you
We
don't
can't
have enough
find
data.
Okay.
Not enough data.
Yeah, that's true. It's okay.
Yeah, I understand.
That means that for instance, Copalinga, we got very, very good record.
Certainly Copalinga has the best. But if you do genus, Bomboscaro is quite good. If you look at genus, Bomboscaro is quite good.
But we also have for Finca and for San Francisco, we have
a couple of them. Yes.
relatively good results.
Some of them, but not too many.
And we can make also that for family.
Yes, I think I
Can
have
we
it.
make that a plot for family?
I think I have it somewhere. I just didn't put it in. Let's see. I think I have it. Let me see if I can show you. Okay, it's this one here. This is for family. Is this bigger? It should open. Oh, here it is. So this is for family.
That one.
So you can see for family, there are quite a few good ones.
It's very interesting. For family, we can find for Copalinga, for San Francisco. Okay, nice. In general, Copalinga is the best one.
Yes.
Yeah, that makes sense because remember that Bomboscaro is the same elevation, but Bomboscaro, the forest is more close.
Yes.
And it's relatively more difficult to record the species.
Yes.
But in Copalinga is a big forest, and the forest in general is more open, and then we could record the birds relatively more easy. Yeah, that makes sense. And the San Francisco Finca, we can find also relatively good results here because they have a lot of species also. The bird species.
Well, you get more. Yeah, but I mean, it's not so much how many species, but how common a species is. I mean, here you have many species, but you don't have good data.
Yeah, that means we have, it is very close to the axis, we have good data, but it's not really clear the difference. That means that because if we are very close to the axis, it's not really good result.
No, it's not a good result.
Nice. Even with family.
Yeah, even with family. So let me go back to this one. And let's see.
Yeah, it's okay. I understand.
Right. Now this one here, whoops. You can say something like that. Yes. This is the plot I showed you. There are several plots like this that are showing different things. This one is showing the confidence interval.
Confidence interval. Okay, that's right.
What this says is that the confidence interval is smallest up here, and it's biggest down here. The confidence interval is like about 50. Even the best is like 50%, but here it's up to 100%. So these down here, the confidence is very small. But these measurements are not too bad. You have maybe 70%, 60-70% confidence interval.
Nice. Okay.
So another one here. This one is. This one talks about the variation.
Yeah, the variation
between the different locations.
And with genius, we have more variation.
Not really. I mean, the yellow means higher variation.
Exactly.
So if you have more points up here, it's lower variation. So genus has more points up here, so it has lower variation. Like this one has low variation. These have lower variation. These have high variation. This has very high variation.
Yeah.
And see here also you have, most of them have very high variation. You have a few that are lower variation.
Yeah, well, we can find by species high variation, and by genus we have less variation.
In many cases, yes.
Yeah. Okay. Very good.
This one here, this is also showing location variation. It's another way of showing it, but it's showing the same thing. Now, then we have these graphs.
Yeah, that plots are the more important plots.
Yeah, this is the most important plot.
Yeah, let me try to explain this.
Okay, sure.
The blue bar means the variation respect to the mean.
Yeah, I have to say uncertainty. The uncertainty of the mean.
There's uncertainty but about the mean.
So the black is your estimate of the mean, but the blue is 90% confidence. So
the blue, repeat please.
The blue is 90% confidence. The confidence.
And 19% the variation.
90% confidence.
And the red one?
The red one talks about the confidence for a point for each individual location.
Yeah, and that is what the delta value is known as delta. I don't remember the name.
Say it again.
But the red one means that we have a lot variation within each region.
Yes, within each region, from one location to another location.
Exactly.
There's a lot of variation.
But each bar with mean is our species.
So, this, I think this is all species. Yeah, this is all species, I think.
And what about the genus?
I think I have genus down here. Genus in.
I think it's genus. Okay.
I have. That was family. Let me see.
Maybe family. Could you show me a family? Yeah.
Family would be. Let's see. Is it this one here?
Maybe that one. Okay.
Yeah, this is family.
Still the variation is very, very high.
Yeah, but the blue bars are smaller.
Yeah, the blue bar, the uncertainty decrease
yes.
with families, but the variation between places in the same region still are
very high. Yes.
very big.
Yes.
Cool. I understand very well. Nice. I think it's very, very nice result.
Okay.
Cool. Yeah. Nice, Christopher. I understand. We have other plot or something like this?
No, this is the last one.
Cool. Yeah, for me it's more clear now. I understand, but I understand the result, but I wanted to understand the formula, the equation.
Yeah.
But now for me it's more clear.
So,
Nice, Christopher.
So,
The next week I will send you the biological explanation of the first manuscript.
sure. Okay.
Yeah, and then I will work in the biological explanation of this manuscript.
Mm-hmm. So what I would like to do with Maria, if she has time, is the hypothesis we have been saying is that that within a region, one species may substitute for another species. That in one location you may have one species which is very common.
Yes.
And then in another location you have another species in the same genus, but it's a different species. And that one here substitutes for this one.
Yes.
So I think we can test this hypothesis mathematically. So if Maria has time, I would like to work on that with her. That to show that the data shows that one species can substitute for another species.
Yeah, I think that's the way to understand that part, because, yeah, remember, I told you the idea is that we have different species in different places,
Yes.
but the role is the same.
Yeah, but you can't tell that from these graphs, because this variation for family is less than species, but that may simply because the family have more observations.
Yeah, of course.
It's like this one has more. This has more absurdity. It may just be because family has more observations. But so part of this is because family has more observations. Part may be because the species are substituting for each other.
That's true.
So we have to do some mathematical analysis to see if this.
And how you can do this?
Well, I would have to work out the mathematics with Maria.
Okay.
But there's a way of doing, you do something with correlations.
Maria Jose, do you have an idea of how to do this? Hey, Maria. Maria, are you still with us? We always have a problem with
Audio, yes.
her computer because it's quite old.
Yes. It's all right, but we do have the recording.
Nice. And the idea could be try to find a mathematic way to find this exchange, this species exchange.
Yes, that's right. That's right. To show this mathematical way to show the species exchange. And I think that would be very interesting.
Yeah, that's good. Yeah, I understand. But do you think if Maria and you work in that part, try to understand how change the species, but maybe the roles are the same, and we can change different species, but the roles are the similar, and with that result, we can start to make the final manuscript.
I think we can start now, because I think we can start now. That because we can simply add that on. I think there's enough here, but that one you would just add something.
Yeah, that's true. I understand. But I think for me it's quite good. I think the results are very, very, very good. But yeah, that I think is a plus for us try to understand that interchange of these.
Yeah. I think that will be important.
That's good. I don't know. Maybe Maria. Let me call Maria. Maybe she can work with you in this.
Yeah, I think she's doing some other projects with with Jaime. But,
yeah, she's working in the project still with us, but she has a short time because she's going to study her master.
She's going to Brazil, right?
Yeah, she's going to Brazil,
or maybe to Italy. I don't know.
Yeah, but she still wants to keep the contact, and she still is working with us in some projects. With Maria Emilia and Orlando got some money to pay Maria, and she still is working with us.
Okay, good. Very good.
Good. Just let me call Maria.
Hi,
Hey, Maria. Thank you